Uh, Assalamu alaikum and good afternoon, all the participants of IICC 2021. Um, we are going to start our session 2 of keynote talks. Uh, Now, our speaker is Dr. Zyta Ziora, currently working in the Institute of Molecular Bioscience, IMB, University of Queensland, Brisbane, Australia. She's a medical chemist by training and also a lecturer in biotechnology program in the School of Chemistry and Molecular Biosciences, SCMB at University of Queensland. I hope that her presentation will enlighten you and it will open new door for mutual collaborations between chemists in Pakistan and chemists in Australia. So, Dr. Zita Ziora, it's a now it's all yours.
Thank you very much. Thank you, Professor Amtul, for your very kind introduction. And thank you for all the organizers to let me present to you and share with you my research that I'm doing right now. Let me share with you my slide. Please just confirm that you can see my slides.
Yes, Doctor. Yes, we can see your slides.
Okay, so let me check if they are working properly, because I see that there is something blocking me to move on, but now it's okay. So the topic today is about improving the skin conditions. But before I will talk and I will share with you my research, let me introduce some facts about the University of Queensland, where I am coming from. This university is located in Brisbane, which is the capital of Queensland state. It is sitting in the center side of the eastern coast, actually, of the Queensland University, which is in Australia. You can see here the very beautiful view of Brisbane by night. This is the building skyscrapers around the river, which is actually with the name Brisbane as well. The other city, which is also well known in the world, it is the Gold Coast, which is situated slightly down south side of Brisbane. And then you can see also very high skyscrapers sitting just in front of the ocean. Another fact about Australia, when the first smallpox vaccination was attempted, Australia was in the very beginning of creating this as a nation without even name Australia at that time, because as you can see from the map, Nuova Holanda, it was called that time. And also, as you can see, the coast on the north part and the south part were not well recognized and characterized at that time. Then in 1936, 3 years before the World War II was happened, the Santa Lucia, the campus where the institute where I am working on is not created yet. There is actually any sign of the buildings for the University of Queensland. Then 10 years later, the first buildings were constructed with the Great Court presented here in the core site of St. Lucia. And this we can call as the beginning of University of Queensland. Now you can enjoy the full view of St. Lucia with the Great Court being presented in the center, and the Institute for Molecular Bioscience, where I am working, is presented also here. Then closer view of this Great Court, the entrance to the Great Court, the institute where I am working, and the School of Chemistry and Molecular Biosciences, where I am teaching as a lecturer. Then closer view to the institute with all of these 4 wings, north, west, east, and south. Then another view about the flora and fauna with beautiful birds, which we have here at the St. Lucia University Lake, pelican birds, turtles, water dragon, and eels. Then let me move to the topic of the talk today about the skin treatment. As we know, all of us, we differ with the color of our skin due to the pigmentation which we have on our skin. So when we have the normal skin structure, as I speak here on this cartoon, we do have dermal fibroblasts, melanocytes, but after skin exposure to the sunlight and the UVA and B, the dermal fibroblasts changed to cancer-associated fibroblasts, and the melanocytes converted to melanoma cells. All of this happening because of the ultraviolet radiation. When the melanocytes trying to protect themselves, they release and they increase production of melanin pigmentation, which initially it is as a sunburn results, but later when the pigments, they start to aggregate, this is the first symptom of melanoma cells. Speaking about global burden of melanoma, you know that the higher risk is in Europe, Northern America, and of course in Australia. When you can compare women and men developing this disease, there are not big differences, except Australia. In Australia, young women aged under 35 are at the higher risk, over 20% than men. And of course, the risk factor, it is the ultraviolet radiation coming from the skin exposure. Melanoma at the early stage, when it's recognized, could be successfully treated with the surgery. But after surgical resection, wounds which are needed to be healed, they have sometimes complications, like, for example, microbial infections. Therefore, we need some post-surgical antimicrobial treatment, and this is what I am working on. Again, about our healthy skin, which we have with the melanocytes, but after skin exposure to the sun, the melanoma cells, they are created, which they need to be removed by surgery. And then there could be the bacteria complications, bacteria infection, which will all cause complex skin problems with this melanoma cells being on our skin. So definitively what we need potential treatment, all to just remove this bacteria growth and preserve the melanocytes looking the same like it was in the healthy skin. In the group of researchers where I am working in the institute, we target covalently modified antibiotics to combat these pathogens, which are multi-drug resistance. We work with the antibiotics which target bacteria membrane. Why? Because bacteria membrane, it is the first shield defense of bacteria. So if we manage to destroy the bacteria membrane, then it will be easier to inhibit the bacteria growth. In 2018, we published in Nature Communication our first paper as an attempt to use our modified antibiotics against bacteria which are resistant to vancomycin and daptomycin. How we did it? We synthesized the modified antibiotic by introducing through the linker a peptidic fragment reached with the positively charged fragments, which could neutralize negatively charged phospholipid head groups. And then by introducing lipophilic membrane inserted elements, which could anchor the whole construct into membrane, it was possible to increase the vancomycin concentration and facilitate the interaction of this antibiotic with lipid 2, which is on the surface of the membrane and which is responsible for maturation of the bacteria membrane. So by disturbing the maturation of bacteria, it was easier to inhibit bacteria growth. Why we are planning to modify antibiotics? Well, we all probably remember the big boom about the golden era in last century, especially starting from 30s years of the last century, when they were discovered and characterized and registered by FDA and other administrations, many antibiotics. But soon after, there was observed antibiotic resistance of the bacteria strands being inactivated to these antibiotics, with the flag example of daptomycin being registered by 2003 by FDA, and then soon later, 2 years later, the first strains of bacteria resistant to this antibiotic were also observed. Therefore, In my research, I focus on non-antibiotic antimicrobial agents, especially looking for the natural compounds and also metals, like in this case, silver, because silver as an antimicrobial agent was used a really long time, 1,000 years before the first antibiotic was applied. You can see from this timeline that Chinese, they were using needles for acupuncture. It was also used for the preventing infections. When that time, even it was not known what the microbes like bacteria and fungi, they look like. It was used for the storage of drinking water, infection treatment. Also for filling our tooth as a cutlery for wound treating, for water purification, and also over-the-counter drugs. Unfortunately, because of the antibiotic boom during the last century, silver was almost forgotten. But now, due to the antibiotics losing their battle, it is the big renaissance of the silver to be used and applied in medical purposes and products. Why? Silver has not only one mode of action. Silver, because of the positive charge of silver ions, could neutralize negatively charged bacterial membrane and can also interact with the peptidoglycan cell wall. And then when it enters the bacteria cell, can also block the cell respiration cycle, could interact with the metabolic pathways, and also could disturb the replication cycle, causing the bacteria death. And because there are 4 modes of actions recognized so far, bacteria are not very easy prepared for the resistance to 4 mode actions attacking this bacteria at the same time. Therefore, we published together with students and other senior colleagues from University of Queensland review about silver as antimicrobial agents in a reputable journal Biotechnology Advances. And soon after, when the article was still in press, I was invited by the science editor from the Times, a London edition, for the interview. And in June 2018, the interview was published in the newspaper, and a few days later was available on the website. Encouraged by this very positive response about our review with other colleagues, I published in Antibiotics another review about not antibiotics that time, but about the silver as a non-antibiotic product in the patent view to be used as a medical application. And while preparing this review, we actually noticed that majority of the patents are written in Chinese, with only 25% being in English. The reason for this is simple. In China, it was much easier to register new patents, and it was for free. While in Europe and in US, when we would like to get US patents, we pay a lot and we wait very long for receiving the registration. I also contacted a Bayersdorf and Nida company to ask about their products, and they shared with me a quite interesting story. They did the survey with their female and male customers, asking who would be interested in their products containing antimicrobial silver. The response was coming positive from men who were more interested in the biopotency of their products, while female, they were more interested in the fragrance than the antimicrobial property of the product. Then I published with another student from Monash University in Melbourne a review about wound dressing, which we can obtain from the natural polymers. We published it in Carbohydrate Polymer Journal, which according to the website is now treated as a hot paper with the highest citation in the field. Then, as I am also working with other metals, with another student who was that time as a student in the Polish-Australia program in University of Melbourne, who got from the Polish Foundation of Science extra scholarship to stay with me at the UQ for 6 months, we published a review about another metal, ruthenium, which is known as an anti-cancer agent, but potentially also possibly to use as an antimicrobial. We published it in Coordination Chemistry Review. This is a really good journal with impact factor now over 15. Then looking closer to metallodrugs as an alternative treatment to bacterial infections. As I have mentioned before, I was working with vancomycin as a glycopeptide, but I did also work with cyclic peptide, aminoglycosides, and beta-lactams. I did formulation of various types of silver, like ions, colloidal silver, and also nanoparticles. And then I applied ITC, isothermal titration calorimetry, all to get thermodynamics of this complex, also to know the stoichiometry. When I knew the ratio between silver ions and antibiotic, I applied these complexes to antibacterial assay to know MIC, minimal inhibitory concentration. And after knowing it, we've done ex vivo study using rodent skin tissue from mice and rats. This was all to check on the agar petri dishes which combination of the silver with which antibiotic was the most active against bacteria which grow on the animal skin. Then we also did similar study using biofilm assay. And then looking closer to the investigation of this complex and interactions in this complex, according to our NMR data, we found out that actually silver is not creating only simple ion pairing interactions due to the positively charged silver ion interacting with carboxylic moiety, which is negatively charged, but also there is the weak spread over the whole molecule interaction of silver with hydroxyl groups coming from carbohydrate, from the aromatic fragments, but also interacting with amine, amide group, methylene groups. So all of these small weakened interactions all together, when we combine, they were the reason of this very strong synergistic effect which we got of these complexes. We also did use inductively coupled plasma ICP for detecting the exact amount of silver in our complexes. And then we did the bioevaluation, as I mentioned before, in vitro, using it against gram-positive and gram-negative bacteria. We use it against biofilm, which is possible to buy commercially, and also we use multi-species oral biofilms, which we obtain from the School of Dentistry. We did the ex vivo on rodents like rat and mice, and also on porcine skin on, using pig ears, and also we use human skin. This was the gift from the dermatological clinic from Princess Alexandra Hospital from Brisbane. When we observed this synergistic effect, we published it in Journal of Inorganic Biochemistry. Then, as I mentioned before, I am also working with the natural polymers. So I do obtain them from the fruits. We know that when we drink very sweet juices, there could be created bacterial plaques on our tooth, and this could just destroy our beautiful smile. So what I did the formulation of the extract which I could obtain from the post-production of the juice leftover, we can say, seeds and peels, mixed it in the formulation with silver ions, all of it to obtain antimicrobial product which could prevent our beautiful smile and also protect our skin. Also, as an example of the collaboration with the industrial partner, who is the producer of de-alcoholized wine, from whom I got the leftover from the wine production samples, which we extracted for several samples. We did also the bioevaluation in vitro against gram-positive and gram-negative bacteria using commercial available biofilms, and also this multi-species oral biofilms ex vivo using the rat, porcine, and human skin. And all of this synergy, when we tested it for the specific fractions, we obtain based on it, we obtain from these fractions our first prototype sol gel product, which we are planning to use to protect the skin, and planning soon commercialize it. In case of the work with the nutraceuticals, like compounds separated from the fractions or extract, I do this work with the students, and then I am very happy to announce here that I do work with the students from Pakistan, like here the example, Mohammad Kamar Salim, who holds now the PhD, and I published with him our research work in Journal of Ethnopharmacology, which was published this year. I do also collaborate with the academics from University of Queensland and with another research partner. This is on silver as a disinfectant, and now we hold the first patent. Speaking about the international collaboration, I do collaborate with the professor from Egypt. This professor is in the Alexandria Institute, with whom I published in Molecules this year, formulated of quaternized aminated chitosan nanoparticles. This is all for the improving wound healing. Another example of the student from Pakistan, Wasim Ulbari, who is from University of Malakand. He holds the PhD for the moment, and we are now submitting the research paper and also the review. Another student who now holds the PhD, Yasir Nazir. This was the collaboration with Professor Muhammad Zaman Ashraf from Allama Iqbal Open University in Islamabad, Pakistan, with whom we published our research work in Bioinorganic Medicinal Chemistry Letter and review in European Journal of Medicinal Chemistry. And with Professor Uzma Yunus, we started another international collaboration with Dr. Paul Hodgson from Durham University from UK. Then together with colleagues, academics from University of Queensland, coming from the same institute as myself, and also from the School of Dentistry at Oral Health Center at the UQ, and from the Agriculture Institute, and also from School of Pharmacy, we created non-formal green vision. The mission of this vision is to bring to market simple yet cost-effective natural products which will show antimicrobial potency and also which could prevent melanoma skin cancer development. This is the scheme what we are planning to get, and we already have the first prototypes which we are planning to commercialize and put on the market soon. Speaking about the publishing, I also would like to announce, like it was announced during the first session, about the special issue. This time it is in the antibiotics. This is special issue about antimicrobial silver in medicinal application. And I would like to encourage all of you to contact me, because I can help you in publishing your research or the review in this special issue in antibiotics. And by this, I would like to acknowledge the collaborative work which I do with all the colleagues, seniors and students and PhD candidates from the institute where I am, from Oral Health Center, from School of Chemistry and Molecular Bioscience, and from the Queensland Agriculture Institute. By this also, I would like to thank all of you for your kind attention. Thank you. I would like to thank. And I think I will stop sharing, and then I am ready for your questions. I think that we do have questions, so let me see.
Thank you. Thank you very much, Dr. Zyta. Very nice presentation. And now it's the time of question answer session. And
yes, I do see the first question from Ayesha. This is the question: what is the main reason for the frequent skin problem? Good question, because we can say that this is not really easy to say what is the main reason. This really depends on where you do live, and also what is your pigmentation as a skin protection. Because as I have mentioned before, this skin problem with melanoma as a skin cancer problem, the disease, it is usually happen to people who have pale color of the skin, means that they have very little of melanin, this pigmentation on the skin. So they don't have this natural defense to protect against the UV radiation when they expose themselves, the skin, on the sun. So melanoma would be, I would say, the major problem for the people with the low level of pigmentation. While for other people who have darker skin, there could be the problem like psoriasis, which is also a problem with the skin, with doesn't regardless, I would say, the pigmentation being present on the skin. The other problem, it could be wound healing, which we have. Like, for example, if we have the tendency to rash and to scratch our skin, or if we are going in the area where there are a lot of bacteria and where this is the high moisture, and the bacteria and fungi, they can attack very easily our skin, not really well protected. So there are a lot of other reasons, which is difficult to concern for the moment and to say that one is the major one. I think that the answer would be depend where we live, and depend what the skin condition we have, because genetically we could have the skin problem and this could be not related to us, regardless of the place where we live. And this could be also related to the environment where we live. Like, as I mentioned, this could be not only the UV radiation, but this could be the high humidity. I hope that I provided the answer on the question. Then the next question, it will be, is the melanoma present in sunlight? I thought that I also explain what the melanoma it will be. So in our skin, we have melanocytes, which are protecting our skin with regulating the amount of the pigmentation, which is melanin. The color of our skin is related to the amount of this melanin. Like people having pale color, they have very little of this melanin, while people with darker skin, they have enough amount of this melanin being already genetically presented in their skin, so they don't need to be worried about the melanoma. This is the reason why melanoma is not really observed in the South America or Asia, because the people have a little bit more pigment in their skin. And also in Asia, like in China, there is a little bit less sun for them to be exposed to. So to answer on the question, melanoma is not in the sunlight. Melanoma is the consequence of too high melanin being produced at the very short time, and especially melanin as a pigment aggregating, because this aggregation could cause not enough oxidation in the skin. This could cause the redox effect. This could cause some other sun-related effect, UV radiation effect, damaging DNA of our skin. And this, as a consequence, could be the growth of the melanocytes converted to melanoma cells. I hope that I also responded on the question. The other, melanoma is effect on only skin or cause of infection. Melanoma, this will be the first and the most important as a side effect of the sunburn and the wound healing, which will be due to the damage on the skin and then a later exposure of the skin to bacteria. So I would say that bacterial infection or fungal infection as a microbial infection, it will be consequence of melanoma, but not the other way. And then the other question about tooth, how cavity attacked our tooth. Cavity, it could be actually related to our hygiene, which we have with our tooth brushing daily, which you know we should do twice per day, morning and before we go to sleep. If we don't keep this personal hygiene, during the day, when we drink a lot of drinks containing a lot of sugar, carbohydrates, and other products,
thank you so much.
Simply sit on our tooth and create this kind of plaques. And these plaques, which are not removed, this could be the source of bacteria to create biofilms. Means that this could be the source for bacteria to create their good environment to live, to control the surface of the tooth, and this could be the source also of the infection. I hope that I answer on the question. Do we have more questions? I think I need to close it and check if there are some other coming. Yes, I think there are coming others. What is the purpose of microorganisms on the skin? Good question. I actually didn't talk about the healthy microbiomes which we have on the skin. So as you can guess, our skin is also the environment for the good bacteria. The good bacteria, they are protecting our skin, creating the good biofilms. They will protect, they live in the symbiosis with our skin cells, and they will protect our skin against the other pathogenic bacteria which could attack us when the skin is wounded and when the sunburn is causing the destroy of the good bacteria. So I hope that this is also the answer on the question. Are there any side effects of silver as antibiotic? Silver is not antibiotic, is as a non-antibiotic antimicrobial agent. Good question. Yes, the silver is still a little bit controversial agent because the very well-known side effect, which actually is not causing big disturbance for our life, it is argyria. This is the presence of silver particles sitting on the surface of the skin and giving this kind of bluish gray color on the skin. And this is the well-known side effect of overdosing of silver. For a long time, it was treated as absolutely not possible to remove a side effect, but now with the technique, especially with using lasers, it's possible to degrade, to break the silver particles being on our skin. And it's possible to release the silver particles to our system and through the circulation going on through our system like blood and gastrointestinal system, is possible to wash out our body from the silver. There are some other reports about silver being present in our organs, but when we stop using silver, and when we do the simple break of the silver treatment, our organism is ready to make the clearance of the silver particles and silver ions. I do hope that I answer on the question as well.
There are a few more questions. Yeah, we are running out of time. Let me read the last question. According to some medical reasons, nanoparticles are known to be effective for human health. Does it depend on the capping agent that it will reduce the effect?
This is also a very good question, which was not possible for me to talk about. This could be that nanoparticles, which are obtained in the chemical way, they could contain, even after purification, a very little amount of this reagents, reducing reagents, and this could be the side effect. So this is the reason why very often now for the green synthesis, it is used the biomass. And this is also what I am planning to do. So this natural product, which I am using